How are our chromosomes really put together? Please take the time to listen to this introduction so you will better understand the video. This information is critically important in our understanding of many kinds of diseases, including cancer. In most primitive life forms like bacteria, the DNA chromosome is composed of one or more circles of DNA. This is also true of many kinds of plasmids and viruses. Many scientists believe we evolved from primitive life forms like bacteria, etc. So it would be natural to conclude that circular DNAs were involved in this evolutionary process. Paradoxically, scientists believe our chromosomes contain only a single strand of linear DNA. They base this on early genetic findings that genes behave as if they are linearly arrayed across the length of chromosomes. They have also found evidence of linear DNA replication in some plasmids, viruses, and bacteria, involving structures called telomeres. Telomeres also exist in the chromosomes of our own cells, which is why scientists believe our chromosomes replicate using only a single strand of linear DNA. If this is true, then it should be easy enough to spread out at least a fairly large portion of a chromosome without breaking it, and confirming this under a microscope, just like they show in textbook models. However, if all that is seen is circles like those shown in the beginning of this video, then Houston, we have a problem. Hi there, my name is Dr. Frank Abernathy. I'm a retired cell biologist graduated from Ohio State University. And I want to talk to you something that's a little different from your usual fare on the internet. No cat videos or politics or anything like that. I want to talk to you about something near and dear to my heart, which is chromosome, and especially their structure, how they're actually put together. Now, when I talk about chromosomes, I'm talking about your chromosomes, my chromosomes, and other kinds of chromosomes from animals, mammals, fish, reptiles, and plants, etc. Now, if you look in any textbook, you will find a very simplified diagram of a chromosome that's been taken apart. And what you will see is I'm going to show you a rubber band models here to demonstrate what I'm talking about. This rubber band, if you think of it as a very long DNA, actually there's some DNA that's pretty much that long, really, in actual reality, 8.5 centimeters. If you spread them out from our cell at chromosome 1. But if you think of this as just a chromosome, DNA, I'm sorry, the DNA itself, you've opened up the chromosome and stripped everything away. They say that it's just nothing more than just a straight piece of DNA. And the reason for that rationale in all these textbooks is because they've known about chromosomes since the 1840s, and even as late as the 1880s, they realized that they are individual entities and that they're linear shaped, they're X-shaped, and they found out throughout the course of the 20th century, they determined that their DNA associated with them, and that the genes seem to line up across, and the genetic information suggests that the genes line up across these chromosomes in a linear fashion. Therefore, they're thinking the DNA itself is lined up in a linear fashion because of that. And I'm saying that that's an erroneous conclusion, and you have been misled by the mainstream science people, and they have tried for decades to come up with a model that allows linear DNA to replicate itself. And so they came up with the idea that telomeres have a mechanism for doing this so that you can replicate a linear molecule of DNA, like the one I was representing with the rubber band. But scientists also know that there's other kinds of DNA that exist as well, and they look more like this, just like this rubber band. It's just a circle. If I could spread it out, it'd be a perfect circle, right? If I could get it to straighten up. And you find this kind of DNA in things like viruses and bacteria. Now, I would assume most scientists would assume that our chromosomes evolved from simpler life forms. And there's not much more simpler life forms than viruses and bacteria. So how do you create a linear chromosome from DNA that is circular in nature? There seems to be a disconnect here. And I would like to talk about that a little bit more if you don't mind. So let me show you some other rubber band models. Scientists are well aware of these things. These are like links in a chain. It's two circles of DNA, but they're not really connected. As you can see, they interlock. They interlock, but you can't pull them apart because of the topology of it all. And they know that these things exist inside cells too. They also know that, now I'm showing this is, they don't really show circles connected like this, but you'll see that there's a connection right here. And if you cut here or cut here, you would create an X-shaped structure. And if you cut here, something else would happen. But let's not cut there right now. If you look at this thing, they would say that's circles that are getting ready to separate. They've replicated, and they're getting ready to peel off from each other. And when they do, you've got two brand new circles, sister circles. So they get done. There's 4 strands in here, 2 from this one, 2 from this one, and they're base pairing. And this can also be involved in recombination, but when they're done, they just separate, and that's it, and they come apart. Now, I'm going to show you something that you won't find in your textbooks. You can find it on my blog, evolution4, but you're not going to find this in any textbooks that I'm aware of. And that's this model right here. Looks very similar to the other one, doesn't it? You know, and they're stuck together. Same kind of pairing. Same kind of pairing. The tetrameric four strands base paired together. But this one's a lot stronger because there may be covalent bonds holding it together. And you can't just peel them apart like you did that other one. And let me show you what I'm talking about. If we were to cut this thing in the middle here, like this, you see, they still stay together. They don't just peel off. These dimples, you got 2 dimples here. You've got a big circle now instead of just 2 little ones. They've fused. Those 2 little circles have fused, and these dimples are holding them together. They're not coming apart. And I'm saying that this kind of a structure does exist in our chromosomes, and they become more prevalent as our cells undergo embryonic development, that you get more and more of these. You can read about that in the blog. Now that's something you're not going to find in your usual textbooks. So not only can they be cut like this, but they can also be trimmed. In other words, you could cut them not dead center, but you could make 2 cuts. You could have cut it a little more here and a little more there, and then there would have been a piece in the middle that would have come out. And then that means that these things have been permanently changed. Information has been lost. Now I'm going to show you some real photomicrographs of what I think is chromatin that has these circular structures in there. And they also show fusion events occurring with dimpling. And I'm going to show you that too. So these are things that are not discussed by the mainstream biology books. And I think it's something that has been sort of really ignored and brushed aside because this kind of data does not support a linear DNA model in chromosomes. So in my next segment, which will be coming up in just a second, I'm going to show you these pictures and the models to support them. Thank you. This is a photomicrograph of mouse cells that are literally falling apart under the microscope. Magnification is probably 400x, 400 times normal. And these large circles are probably the remnants of the nuclei, although they're not all the same size. And if you'll notice, you'll see a pouring out of little tiny circles, many of them with little beads around their circumference. And it's actually a very beautiful micrograph. And so this is the beginning stages of the releasing of circles, peeling off of these little circles off of the main, somebody called it my advisor called it a mothership, where they're just shooting out like little spacecraft from Independence Day. Some of them even have little circles inside of the circles. So and they're all different sizes. So they're just now being released from the nuclei as this superstructure is being revealed, to my best of my knowledge, for the first time. Now in this slide, you'll notice that the circles, you don't see very many large nuclei-like objects anymore. Most of the circles have been released, and they're starting to get larger in size, though there are many, many smaller ones that you can see all mixed in together. But they're basically separate circles for the most part. In this next slide, you can see the arrow, yellow arrow, pointing towards two little circles that are dimpled, that are fused together. These things have gotten bigger and they're starting to fuse together. You can see a whole lot of these things. Some of them even have little junctions still within them. This is a magnified portion of the previous slide, and I've included some more arrows so that you can see more of these dimpled structures in greater detail. This next slide are electron micrographs of DNA, purified DNA that was isolated from these cells. When you look at the top picture, these are submicroscopic. You couldn't even see these under the microscope that was used to show the other circles. These are very, very small. One of them shows two fused circles at the top, and the other two below that show the dimpling effect even at this level, at this very microscopic level. So there is a range of these things from huge all the way down to submicroscopic. Okay, we're going to switch back to the light microscope. This is a picture of something at 400x, and that has been magnified a lot by blowing it up. And you see a circular structure with little blebs coming off of it, peeling off of it, of a scaffold. And this is done with phase contrast microscopy. And so it looks like the little circular elements are leaving the scaffolding. And this seems to be cell cycle dependent, because a lot of these cells may have been in asynchronous replication, which means they were all in different phases of this cell cycle. And you can see evidence of this in the next slide. Okay, this is a fairly complex slide. It's kind of hard to say where to begin, so we'll start with figure 29. You'll notice that in figure 29 on the left, that the elements that are encircling the hub there have started to fuse together, and you see evidence of that kind of in 30 and something like that in 31, and especially in 32. It's like those little circular elements, those little mini circles, or microcircles I should say, have started to fuse together. And 33 through 36 seems to be the scaffolding remnants left over when these things depart the large circular element because of disruption in the cell cycle when these things have been isolated on a microscope slide. 37 is another interesting one where you can see the little circular elements, the microcircles with their inner hubs, and you can see one of those in 38, which was a blow up of 39, which the arrow shows that. And they look like grapes ripe for the picking, and they're just ready to come off like they were in the slide that we showed earlier. So you see various and sundry forms of these things, and they look like they're trying their best to form chromosomes. And especially when you get down to 46 through 51, those look like chromosomes, and some of these elements seem to be attached to scaffold, which is when they release from this scaffold, you see this kind of washed out scaffolding like you see in 44 and 36 and 35 when these elements have left the scaffolding for whatever reason, because of interruption of the cell cycle, because the cells are dying under the microscope. And you see there is a lot going on here, a lot that needs to be studied, and it is not being studied. So there's lots to learn about chromosome substructure that we have barely scratched the surface. Okay, this is one of my models to try to explain what's going on here, because what you're seeing is these circles fusing with each other, or they're being released from each other. So you're having two kinds of processes going on at the same time. So what I've drawn here, if you look in the upper left hand corner, replicon 1 and replicon 2, they're supposed to be identical. And you see that tetrameric base pairing from the two strands of 1 and 2 coming together, base pairing of DNA like you see in B. And all of this is reversible. Remember when I showed you those rubber bands that they could peel away from each other when they were done replicating? So now you go to C, which is also reversible, and they are fully tetrameric base pair. That is, they either pair with their sister strands, or they're pairing with the non-sister strands. It's a mix-up so that all 4 strands are interlocked by base pairing. Now you'll notice from when you go from C, you can either go to D or you can go to E. Now you'll notice that when you go from C to E, that's reversible. The arrows go in both directions. But when you go from C to D, it is one way. And the reason that is because, remember when I talked about trimming away those dimples in those rubber bands earlier? Well, that has been trimmed out. There's been a section removed from the middle of those two replicons, and you've lost information. So you cannot reverse that back to C again. Whereas with E, if you just cut it single cuts in the middle like is shown there in E, that could come back together and reform C if you religated those strands back like they were. And in the case of E, it shows if they fuse completely, you get a double circle like you see in F, which can also revert back if you haven't lost that middle section like has happened in D. Alright, in this slide, you're seeing that the circles continue to fuse together, and the beads are getting smaller all the time that go around the circumference of these things. They're starting to get irregular shaped because they're so large, and you're just continuing to see fusions occurring to create these larger and larger circles. Finally, the dye, the acridine orange dye that was used to stain these so they would show up under UV light, is beginning to just fade away, degrade, and you'll notice that these circles have gotten huge. They've just formed this tangled mass of one gigantic circle that you can see there, sprawling all over the photomicrograph. Okay, this is a very complicated diagram which is available on the blog. I'm not sure how well you're going to be able to see it here, but we're going to just talk about a portion of this diagram, the one on the right hand side. That's supposed to represent a complete chromosome, such as would be found in our kinds of cells. So what we're going to do is we're going to split this chromosome in half at the centromere, which is all of those little black tetrads down there toward the bottom. We're going to separate that and just look at the upper half. All chromosomes have what you call arms, and a single chromosome that's not been replicated has two arms, an upper and a lower. We're going to look at the upper arm, and that arm is also called a chromatid. Okay, this is the upper half of that chromosome that was in that rectangle. We got rid of all the verbiage and everything else and put in some new verbiage, made it larger. Hopefully you will be able to see this. If these are hard to see, they are on the blog, all of them. This one's not yet, but I might put it up later. But this is the upper half above the centromere, and it is one chromatid, the whole thing. So if you look at it, it does look linear, just looking at it. And the little, if you look at the little hot dog-like things, one of them says it has a telomere or a linear end. That's just to placate the mainstream biologists so they don't have a fit about it. But actually there's what they call 4 subchromatids. That the chromatid is made up of 4 big circles that are kind of packed together. They look like hot dogs, but you'll see that they have fragile sites between them where the 4-strand base pairing occurs, like we talked about earlier. So and the covalent bonding, so that it's a strong bond, but it's still fragile. They could actually separate or fuse. So those things could actually separate or fuse, and might be the hot spots that exist in chromosomes when they are damaged by drugs or x-rays, and they break loose and they give off circular elements and things that are not associated with the centromere. Now when they're not associated with the centromere, they cannot be separated into daughter cells, and they may or may not be lost in subsequent replications. So that's why it's important for all of this stuff to be attached to the centromere so they can be pulled apart at metaphase. But anyway, so these subchromatids, I call them mini circles relative to the microcircles. It's just an arbitrary term. They're bigger than the microcircles, which you see, they call them condensed microcircles. All of those little tetrads, they look like clover leaves. Those are actually little tiny circles. Those are where the genes actually are. That's the business end of the chromosome here, because when those things either come loose or they fuse, when they fuse, they delete that information I talked about earlier. It opens up an RNA transcription site, and gene expression can occur at that point. So that's what those things are. And the next couple of slides that I'm going to show, they may be hard to see, but what I'm going to show is I'm not going to show these things coming apart as much as I'm going to show. They do come apart, but I'm going to mainly show how they fuse together to form giant circles like you saw in the photomicrographs. So that's going to be the main thing, but there will be some dissociations that occur too. So We will show that in the next slide. I apologize if this slide is hard to see. It is on the blog. What you're seeing in A on the left hand side is the whole chromosome that was shown before, and we're only showing that upper part where it's bracketed, which is the upper chromatid. So what's happened in B with that chromatid is that all of those subchromatids, all four of them, have fused together to form a giant circle, and all the little tetrad cloverleafs are still compacted. And then in C, the cloverleafs open up, and they look like paddle fans. They have all opened up, and you see there's four of them associated with an inner circle. When you go to D, all of those paddle fans fuse together and form the circles that you see down in D, and then those things eventually fuse with the larger circle to form what you see in E, which is actually almost is going to become a giant circle or a big circle with smaller circular elements, which we'll see in the next slide. Okay, now in this slide you see F at the top. Further fusions have taken place, which has also caused some other circles to form. And eventually you get down to G at the bottom, and what you have is one gigantic circle, and then you have smaller circles associated with it. There's 5 bigger ones and 3 smaller ones. So there's 9 circles in all, and all those little tiny dots are where the origins of replication were at one point when all of these things started fusing together. Okay, here is a link to the blog and my email contact information if you want to ask me any questions. The research here was all performed at Ohio State University, and you can find links to my dissertation, manuscripts, and references. And there are lots of references, so I hope that you will take the time to run over to the blog and learn more about this information. Thank you very much for watching this video.